Thank you very much for the kind introduction, and particularly for the OSHA recommendations. So, how many of you have been bitten by a tick? Raise your hand. Okay, just about everybody. So, if you were you bitten around here on Long Island? Yes? Okay, so I very much hope that you found that tick within 24 hours and pulled it off. And that if you didn't, that you got treated with doxycycline promptly, because Lyme disease is no fun. And Lyme disease, of course, isn't the only thing you have to worry about. So, ticks have 3 life stages. They bite 3 times in their lives. They go from larva to nymph to adult. Now, larvae are never infected with pathogens. And that's just because ticks are born uninfected. Nymphs are another story, and adults still more so. So, would anyone like to guess what fraction of nymphs are infected with Lyme disease here in Suffolk County? Pretty close. You're looking at 21% of nymphs and 67% of adults. Now, the adults, thankfully, you usually find them, and pull them off well before the 24-hour mark, which is required for transmission. But, of course, Borrelia burgdorferi, Lyme disease, is not the only thing you have to worry about. Babesia microti, babesiosis, it's commonly associated with Nantucket, which has the highest rates of it. But interestingly enough, this paper, Ian Lipkin's group, just showed that it is nearly as bad here on Suffolk County. That is, there are half as many cases of babesia as there are of Lyme disease here. Nor is that the only one. You also have anaplasma and a different form of Borrelia. And the scariest of all, powassan. Now, these are all fairly nasty rates, but what they have in common, even though they're very, very different from one another, okay, the Borrelias are pretty similar, but otherwise you have bacteria, you have protists, and they're all transmitted to the tick primarily from a white-footed mouse. That is, usually the larva bites a mouse, and if that mouse is infected, then that tick almost always becomes infected. That's why the mouse is the primary reservoir. Similarly, if an infected nymph bites a mouse, the mouse almost always gets infected. The reason why there's a tick-borne disease problem here is because there's a lot more ticks than there should be. And that's because there's a lot more deer than there should be. The only major predator of deer around here, of course, is the automobile. And every deer has on it typically 1,000 or more adult ticks, all of the females of which will lay 2,000 to 5,000 eggs. So every time you see a deer grazing peacefully on the lawn, then you can think of that as 1 million ticks in the next generation. Question? Yes? Yes, the typical deer has 1,000 adult ticks attached to it. So lots of deer means lots of ticks. And lots of ticks means lots of opportunities to transmit disease from one mouse to a tick to another mouse. And, of course, there's also a lot of mice. There are many more mice than there should be. And that's because there are lots of things that a tick could, in principle, bite. Obviously, they're happy to bite anything that is typically small and warm-blooded and tasty. But the mouse is the best reservoir because it's so efficient at transmitting all these pathogens. And right now, the mouse has outcompeted most of the other little rodents in this area. And that's because the mouse is best adapted to the edges of forests. And so what we have done, of course, is we love trees. So we plant lots of trees, we encourage forests, but we carve them up with roads and houses and so on. So you maximize the perimeter of forest. And so you favor the mice over everything else. So you have a perfect storm. We have lots of ticks, and we have lots of mice, which are the best reservoir, which means you have lots and lots of infected ticks. Now, here's the worrisome part. I mentioned doxycycline. So that's the most common treatment for Borrelia and Anaplasma and Miyamotoi, the other Borrelia. Works for all of them. The problem is, here in particular, where you have such a high chance of babesiosis, it doesn't work on babesia. Treatment for babesiosis is something different. And so your physician would have to catch that as being something different, not Lyme disease, like they tend to assume, that it's actually babesiosis, and give you the different treatment. And babesiosis, mind you, is by all reports, nastier than Lyme disease. And then, of course, there's Powassan. So it is super rare, thankfully, and you might think, well, it's in only 2% of adult ticks, and so you don't have to, and so you shouldn't really have to worry too much because you find an adult tick and you pull it off within 24 hours almost always, right? I mean, they're big enough that you notice them, right? Well, all of these others require 24 hours to be transmitted from the tick to you. Powassan's a virus. We're talking maybe an hour. What's more, there is no treatment currently for Powassan other than going to the intensive care unit and them supplementing your every organ system to keep you alive until the virus takes its course. Mortality rate is about 10%. Now, there's only been a handful of cases every year across the entire country, but still, 2% here. That's a terrifying number, given that we don't have any options there. So this is a problem, right? We have changed the local environment so that kids can no longer run freely through the woods. And we don't have a ton of other treatment options. So there is a vaccine against Borrelia burgdorferi for Lyme disease. However, it was pulled from the market a number of years back, the very beginning of the anti-vax scare. And it was only 75% effective in terms of protection against Lyme disease. And it also was never approved for anyone under the age of 15. So you couldn't get it for your kids. You can still get it for your dogs, mind you, and you should, because it works. But you can't get it for people anymore. So there is no vaccine, and there is, again, there's never been a vaccine, nor is there even one in the works for any of the others. That means you should always permethrin treat your shoes and your boots and your clothes if you're going out in the woods, and you should always do tick checks. But it would be lovely if we could actually have kids run freely through the woods. I am a huge Calvin and Hobbes fan. What does Calvin spend most of the comics doing? He's running around in the woods. That's what kids should be doing. And they can't do it anymore. So how can we fix that? Well, we hope that we might be able to prevent tick-borne disease, potentially all of these tick-borne diseases, by heritably immunizing the local mice, because it's the mice that infect most ticks. The mice are the single species that is the weak link in this cycle, except for the ticks, which we'll talk about is probably not a good idea. If we were to release resistant mice into the population, then that would introduce immunity. If we introduce enough mice, or engage in some genetic shenanigans, then we can ensure that all of the mice are immune. Now, what's interesting and compelling to many people is that we can actually do this without introducing any foreign DNA into the mice. That is, we can take native mouse genes, work with that DNA only from a mouse, and put it back into the mouse. And that really matters to a lot of people. Scientifically, it doesn't matter, but it matters to a lot of people. And this is a technology for altering the shared environment. So it must only be done with the consent of the communities. In fact, we need to go further than that. So the way it would work is that some white-footed mice naturally express antibodies against both ticks and the Lyme-causing spirochete and the other pathogens for that matter. Because mice are just like us in that they have an adaptive immune system. And when you're exposed to a pathogen, then the immune cells in your body undergo a competition, an arms race, trying to recognize that pathogen better than all the other immune cells. And they get to replicate more the better they are at doing that job of flagging that pathogen. And so over time, a matter of days, you eventually end up with B cells that can produce very tightly binding antibodies to whatever has infected you. And that's typically why you get a cold for a few days, and then you get better, because your body has learned, it has actually evolved new antibody genes that recognize that particular pathogen. So our proposal is to take those antibodies and ensure that mice express them from birth. Because normally, if you evolve resistance to some pathogen that has affected you, then your children don't get it. It can't be passed on. We hope to fix that, at least for the mice. Now again, this is a proposal to edit the common environment. And that means it should only be proposed if there are clear and obvious benefits to everyone. Discussion should always come before experiments are run. Safeguards have to be agreed upon early. And frankly, if you want buy-in these days, it better be a non-profit enterprise, or you're going to get a lot of suspicion just that you're out in it to make a buck. What's more, new technologies, right? Who's the last person you want to test a hypothesis? the person who proposed it. Similarly, the last person you want to test a technology to see if it's a good idea, if it's working well, if it has side effects, is the person who invented it, meaning that our team cannot be the one to evaluate whether or not this is working, or whether there are any ecological side effects. We need an independent team of assessors. And the science needs to be open and responsive to the community from the get-go. So this project will only move forwards if embraced by local communities. And furthermore, it's not going to replace all control efforts. That is, the earliest possible deployment is 7 years away, and that's not even here. So don't stop with your permethrin-treated boots and your tick checks. I'm a biologist, or really an evolutionary engineer. And biology is fantastically complicated because evolution-made systems are spaghetti code. And the task of biologists is to figure out how all of that spaghetti code works. And we're not very good at predicting side effects. And that's true at just the cellular level when we tweak something. It's even more so at the ecosystem level. And so my general 2 rules for engineering complex systems, no matter what they are, is you want to make the smallest possible change capable of solving the problem, because you never know what else you're going to mess up. And you should always start local and try to test it in a small area before scaling up to the entire system. And so that's the philosophy we're trying to use. In order to do this, of course, there's all sorts of laws and regulations governing what you can and cannot do in the environment. And that's true at the federal, state, and local level. You have to get regulatory approval for all of those. Personally, I'm not sure how useful that is, because the level of scrutiny that I think it should be required for this, the level of community involvement, should be much greater than the regulators are going to apply, including ethical analysis, which the FDA throws out the window by law. And in terms of starting local, uninhabited islands first, then inhabited islands, and then maybe the mainland afterwards. And it's important to emphasize that for now, we don't want those changes to spread beyond the islands. That is, if you're running a field trial, you want it to stay confined on the field trial. So we're not using any form of gene drive, although we'll talk about that later. So, as we mentioned earlier, this is an ecological problem. All these pathogens are passed between the mouse and the tick. And we wouldn't care at all, except for the fact that if we get bitten, then we get sick. Interestingly enough, the mice don't get sick. They don't get disease at all. They've been here for so long, co-evolving with the ticks and these pathogens, that there is, as best we can tell, no effect whatsoever when a mouse gets bitten by a tick and infected with these. But we do, and so do our dogs, because they're like us, they're invasive here. We're too new. But what if the mice couldn't become infected at all? Then you would break this cycle of transmission, and of course, if you didn't have any infected ticks, then you wouldn't have any infected people. So can we do that? At least, we probably can't get all the way there, because I mentioned that the mice are the best reservoir, but they're not the only one. So, but if there were no infected mice, there would be very few infected ticks, and therefore very few infected people. But it wouldn't go to 0. How far it goes is going to depend on the ecosystem. So would this work? Like, do we have a hope? Now, there's not a lot of data on this. We have 2 studies available. A group of saints went out there into the wilderness, in the forest, and they light up mouse traps. And they took the vaccine, and every mouse that they trapped, they vaccinated it, they tagged it, and they let it go. And they did this for 5 years. And they reduced the infection rate for Lyme disease only, Borrelia burgdorferi, by 42%, and the tick infection rate went down 25%. But of course, this was done on the mainland, in Dutchess County, and so there are always mice moving in and out of both the experimental and control plots, and there's natural variability, so they couldn't control for any of that, and they couldn't get all of the mice either. They know they couldn't get all of the mice. But still, not bad. It suggests that it would work. The second study used this oral vaccine, and the idea being that if you just leave out bait, that if the mice eat the bait, then they become immune. Would be lovely. It's actually a recombinant living bacteria expressing this. And they reported 25% reduction in tick infection rates after 2 years and 76% after 5 years. But when they actually tested the antibody levels of these mice, it was barely statistically significant. The vast majority of mice were not mounting an immune response. And when you look at the relative background infection levels and the history of infection levels in their experimental and control plots, it just so happened that the experimental plots had started out when the study began at a really high, unusually high level, and then it just declined naturally over the course of the study. So given that their vaccine wasn't actually doing very much, and the other papers and literature suggest this study really isn't very good. So we're left with one study that suggests that this will work. But it's a pretty good study. Not much, and I think we need to try something. So we have two options. Number one, Lyme disease is the biggest problem of the tick-borne diseases. CDC estimates 300,000 cases across the United States every year. So we could just immunize these mice against Borrelia burgdorferi, the Lyme disease pathogen. And that's actually very easy because we already have a vaccine. And the vaccine is against a molecule that is pretty invariant across all of the Borrelia strains. So it works for most of them. But of course, it would not prevent other tick-borne diseases. Be very precise and targeted to Lyme disease, very unlikely to cause any other ecological effects because it would just hit that one pathogen and nothing else. But it wouldn't do anything for the others. The other option is to immunize the mice against ticks. Because when the tick bites you, it has to have some way of tamping down your immune response. Because when it's attached to you, it's linked into your bloodstream, and unlike a mosquito, it stays there. And so your immune system will actually respond to that. It will start attacking the tick's cells, because it has direct access to them, of course. It's in your bloodstream. So the tick has to secrete proteins that dampen down your immune response. But it turns out that if you are immune, if you have an immune response to the proteins that are that invisibility shield that the tick injects into you, and you recognize and destroy those first so it can't establish it, then your immune system will attack the tick so ferociously that the tick will fall off and usually die. So there are some very lucky people that have this naturally. So my friend and colleague Sam Telford is one of them. He's been tramping around as a tick ecologist for 30 years. And he doesn't have to worry about anything other than powassan, because when the tick bites him, a few hours later it falls off and dies. So he has a natural form of this. We could do either. But if we do the latter, it will reduce the tick population by a lot. Now, normally when I go in front of a community and say, this is a drawback, then I get a lot of incredulous looks, like, this is a drawback? You're going to reduce the tick population by 90%? How is this possibly a bad thing? But the ticks might play some sort of ecological role. To be fair, every tick ecologist I've talked to believes that we could drive the species extinct and the rest of the ecosystem would not notice at all. But it is possible. We don't understand ecosystems that well. So how would this work overall? Take mice in the lab, vaccinate them against either Borrelia or against tick saliva. So then you have highly resistant mice that have evolved antibodies that make them resistant. Sequence those antibody-encoding genes, and then make the recombinant antibodies in the lab. We're running what's called the yeast surface display to isolate the ones that are the best binders to Borrelia or tick saliva. And then we take the genes encoding those particular most protective antibodies, and we can put them in the mouse genome. Just engineer them into the germline of into the reproductive cells of the mice, so that then they can be passed on to future generations of mice. So then future generations of mice will just be born producing those particular highly protective antibodies, and they will be born immune. And we actually know this works because we've done it in lab mice for completely different reasons. And we also know that there's a very promising vaccine field of research that uses gene therapy, takes already known highly protective antibodies, and injects them in your muscle cells, the genes encoding them, so your muscle cells, which don't normally produce antibodies, they're not immune cells, but in this case they start producing antibodies like they were, and you then are protected. It works great. We can actually, particularly in mice, it works against malaria, HIV, influenza, hepatitis, works against all of them. So it's a very well-established approach. It just hasn't been done in a heritable sense. So once we have what we believe are immune mice, and they test as immune in the lab, we need to run a field trial. So uninhabited islands. We're looking at a variety of different islands off the coast of New York, Connecticut, Massachusetts, and all the way up to Maine. And there's a number of different candidates, but we want ones that are a reasonably good ecological match for the place where we're really interested in looking at. And in this case, that's the islands of Nantucket and Martha's Vineyard. And that's just because they are about the right size that this could conceivably work. And more on that later. Once there's this independent study of whether or not this works on the small uninhabited islands, then the independent assessors, the Data Safety Monitoring Board, would then deliver a report to the communities, which would then have town meetings and vote as to whether or not they want to release mice on their islands. timeline here, we give ourselves maybe 2 years to engineer immune mice. This is an engineering problem, it's not a science problem. We know we can do it. It might take longer than this, but that's our best guesstimate. Then we need 2 to 3 years just to breed up enough resistant mice for a small island trial, because you have to go from the handful of ones that have the relevant genetics in your lab, you gotta breed up enough of them so you have 1,000 or more mice to release on the island. So one small 1 square mile island needs about 1,000 mice. And then we need 2 to 3 years to evaluate the effects and also to raise enough mice for a large island. Now, the large islands are going to be a lot of mice. Up to 100,000 mice released on each island in early spring. Now, you might say, this is crazy. There is no way that any community is ever going to vote to release genetically engineered mice, 100,000 genetically engineered mice on their island. And yet, because this is a problem that people actually care about and actually impacts their lives, and because they are in charge, not us, they're actually very interested. What's more, the introduced mice would increase the local mouse population to at most 200% of normal. That is, we want to make mice that have several copies of the antibody cassette, meaning their offspring will inherit half as many, but will still generally inherit at least one. Meaning we can release about as many mice as there are there, which is normally about 100,000 at the end of winter. Most of them have starved, because nature is cruel. But by context, there's usually 800,000 mice at the beginning of winter. So we'd be raising it from 100,000 to 200,000, but the max is 800,000, so that's actually not that much. I mentioned that they're in charge. Both islands have now appointed members to steering committees, and it's the steering committees that are actually running the project. So they tell Joanna, who is the project manager, she's a graduate student in my group, who's also a resident of Martha's Vineyard, what to do, and she's running the research. So right now she's running the yeast surface display to pull down the antibodies against the Lyme pathogen. And we're talking with NSF about appointing a data safety monitoring board, which would be essentially the independent assessors to look at the data from the island trial, and also from our results in our lab, and just to make sure we're not making things up, not biasing, diluting ourselves. We have a lot of great collaborators. Sam Telford is the one I mentioned, who is apparently totally immune to ticks. And so he's the leading tick ecologist on the islands and mainland nearby. And Lyndon Hu is a MD at Tufts, who specializes in Lyme disease. And Ryan's an expert in yeast surface display. Aurelie is in Lyndon's lab. On our actual research team, Jean-Tine is our ethicist, because a major interest of ours in this project is how do you develop a technology like this, one that's designed to alter the ecosystem, and how do you do that together with a community? We want to figure out a model for how this can be done, because biotech is advancing so fast that we can do this more and more. This is going to be more and more of a problem where we need to decide whether, when, and how we're going to change the environment. And that's a collective action problem. It's not a matter of going to your doctor and deciding whether or not you want a treatment. Informed consent just doesn't work in this case. It's not the appropriate model, so we have to figure that out. And then again, Joanna is the project manager, and John and Devora, 2 other grad students in the lab who are working on it. Now, decision points for the community, because again, the community is in charge. So after we make the mice, then we deliver all of our laboratory data to the Data Safety Monitoring Board, and they need to give a recommendation to the communities. We would need regulatory approval. The steering committees would apply for that. Could be FDA, could be EPA. You might say, why would the Food and Drug Administration have any kind of authority whatsoever over an environmental trial? That should obviously be EPA, right? Well, recombinant DNA, even if it's from the original organism, if you engineer the genome, that DNA that you introduce, that's a veterinary drug, legally speaking, because it changes the traits of the organism. So the FDA might actually regulate the mice. However, if we do the anti-tick version, which is what, by the way, the community is overwhelmingly say, if you're going to do anything, do both. And the anti-tick version will reduce the tick population. Therefore, it's a pesticide under EPA. So it's very unclear which one of them will actually get it. We would need regulatory approval, and of course, the approval of whoever actually owns or governs the uninhabited islands that we eventually select. After that trial, then again, you would have a recommendation and report by the Data Safety Monitoring Board, and then we'd need regulatory approval for the Vineyard in Nantucket, and then they would vote on it. Funding. This is obviously key. If you accept funding from someone, you might be biased by them. They have some sort of control over it. So far, MIT Media Lab, where I work, has provided funding just because my lab is new. We've only been around for less than a year and a half now. And so we're using my startup funding. We've been recommended for funding for a Tick-borne disease research award, which should hopefully cover us for the rest of the laboratory development. And we've had a gift from the Rainwater Foundation, which has some activities on Nantucket. Field trials will require philanthropic funding, because as you can imagine, raising and releasing 100,000 mice is an endeavor. And then, of course, running a field trial and tracking it and all of that jazz, that's going to be expensive, so we're going to need philanthropic funding. All project finances will be transparent, just again to mitigate any potential conflicts of interest as much as possible and ensure that everybody knows everything that is going on with the project insofar as is available. And in fact, we're currently working on making all of my laboratory's finances transparent, except for individual employee salaries, obviously, as much as possible. So this idea really, I just wondered, well, why don't we encode antibodies in the germline for heritable resistance a couple of years ago? And so we called a workshop where island community leaders and scientists, including ecologists, and actually representatives of environmental NGOs, Nature Conservancy and some of the others, and ethicists all came together at MIT, and we just said, if we think we can do this, how should we go about it? And so we sort of laid out the protocol for moving forwards. In June last year, we met with the Nantucket's Board of Health at an open public meeting and pitched the idea and said, is this something you are at all interested in? Because if not, then we're not going to do it. But we haven't done anything to date. We haven't gotten funding. We haven't done anything. And they were really interested. And then the next month, we went to the Vineyard and did the same thing. And as early as July of last year, the Nantucket Board of Health said, okay, we're going to appoint a steering committee. Let's do this. And by May of just last month, the last town on the Vineyard agreed to appoint people to their steering committee. So they're at least on board with having the project continue, which I think is pretty remarkable. Again, 100,000 genetically engineered mice. And we've finished vaccinating the mice. We've sequenced the white-footed mouse genome, which hadn't been done before. It's going to be the first engineering of the white-footed mouse that anyone has ever done. We've made the antibody libraries, and Joanna is even now performing the selections. So this is happening. And this is an example of what we want this to be. That is, for a new technology, we really have to develop it to focus on the most urgent needs. Biotech, when it comes to public opinion and perception of biotech, the reason why you think 100,000 genetically engineered mice, no way, no one will ever do that, is because nobody trusts Monsanto. All GMO foods, all that mess is because it was developed for what? To make farmers a little bit of money and to make Monsanto and their ilk more money. And to consumers, yes, it produces slightly cheaper food, but very slightly, not so much that you're going to notice. There is no obvious benefit to anyone from standard GMO crops. And what's the most common GMO crop? Oh wait, resistance to an herbicide. In other words, they're just making the crops resistant to a nasty toxic chemical that then they can spray more of on. That does not sound like much of a societal benefit to me. And that was the introduction of the technology in the public eyes. This is why people don't trust genetic engineering, because they don't like Monsanto, and that's tied up in all sorts of other societal controversies, not just is natural good, but industrial agriculture, vision of the pastoral farming, etc. So if you're introducing a new technology, it needs to be something the typical person on the street, you say, we're trying to do X, they need to say, that's a great idea. And if that doesn't happen, you probably shouldn't be doing it. That should not be the initial example of the technology. You gotta openly share everything before experiments begin. This also just means more people will have a chance to share their concerns. And it's very possible, again, complicated ecosystems, we don't know what necessarily everything. Someone might think of something that could go wrong that we haven't thought of. And we could check for it, and we're more likely to be successful. This is how science works. You know, there's no scientific method, it's just incentivizing people to check your work. We should do that beyond just the scientific community. Request community guidance from the outset. Again, ecological technology, shared environment, community needs to be in charge. Independent assessment, stay nonprofit as long as possible. You may not be able to scaling up. And start locally. So that's what we want. But all of you are now saying, well, that's lovely, but I don't actually live on the Vineyard or Nantucket. What does this do for Long Island? Well, I mentioned CRISPR for genome editing. CRISPR is a tool that lets us edit any DNA sequence of the genome very readily. In effect, it's a molecular scalpel that can be programmed to find and cut any DNA sequence in the genome. And CRISPR components consist of a nuclease enzyme, which actually does the cutting, guide RNAs telling it where to cut. Now, when we first demonstrated CRISPR in the laboratory, engineering the genome, we used 3 separate pieces of DNA, one encoding the enzyme, one encoding the guide RNA telling it where to cut, and one encoding the DNA that we wanted to insert, the edited version of the gene. And you put all those in the cell, it makes the CRISPR components, cuts the target, and inserts the desired change only. But a couple of years back, I wondered, well, what would happen if you put it all together? That is, what if you made CRISPR genome editing a heritable trait in and of itself? So you put all 3 of them together into the cell. CRISPR cuts the target. The cell repairs the damage by finding a similar sequence, in this case, the one you introduced. And now this reproductive cell of this mouse has everything it needs to do genome editing on its own. This cell will begin producing the CRISPR components because it integrated the genes encoding them. So it's going to cut the other chromosome and edit the other copy of the gene. So now you have 2 copies, which means that when this mouse mates with a wild mouse, since there were 2 copies in this parent, all of the offspring are going to inherit one. And in this offspring, genome editing happens again. 1 copy to 2 copies. And these offspring then all inherit, and editing happens again, and they mate, and editing happens again, until eventually you've released a find and replace element into the population. You give it enough generations, it will find and replace every version, every copy of that original gene, and replace it with the edited version. This is called the CRISPR gene drive. It's a way of spreading a change through a population, even if it doesn't help the organism reproduce. Gene drive systems are ubiquitous in nature, right? From a gene's eye perspective, you want to be as frequent, as abundant as possible. This is a great way to cheat standard inheritance. And nature has evolved many, many, many ways of doing it, including one that works exactly this way. They're extraordinarily common. But with CRISPR, we can do this deliberately. And that makes it very different. Here's the problem. The weakest reported gene drive system to date, so we first told the world that this could happen. We published a paper before we tested in the lab very deliberately, thinking that people should not test this until you describe it. And then tested it in yeast, particularly the laboratory safeguards we thought were necessary, and showed that it worked. Since then, people have done it in fruit flies and a couple of species of mosquitoes, and we've done it in worms very recently. The weakest reported gene drive system is one that, instead of always copying itself over the wild-type gene, it only does it 50% of the time. And it's not engineered to be evolutionarily stable. That is, the cell can just jam the ends of the DNA together, create a mutation. And that mutation then prevents further cutting by the drive system. So if you allow it to do that, and there are ways to prevent that, but in this version they didn't, then it's just going to create resistant alleles that block the drive. And they're less costly than the drive system, so they will outcompete it by natural selection. So we modeled this drive system and found that it still, even though it's the worst one reported to date, the mosquito ones are copied at 98% of the time, the yeast 99.99% of the time. But in this fruit fly one that's only 50%, it still rises to above 30% of the population before going down. And how many organisms does it take? This was a fruit fly drive. If you were to just release them in the wild, how many does it take before you're likely to get up to that peak? Well, mean and median give you different results, but 2, and you're about 50% likelihood. 12, and it's near unity. That's really invasive. So just how invasive? Well, we can actually plot homing efficiency, that is, how frequently is the drive copied. So 50% drive is here, the mosquito ones that work, yeast ones that work really well are up here. And then track the migration rate from the initial population it's released in to whatever population that's connected to. That is, how much gene flow is there between them. And you can have really, really low migration rates, 1 in 1,000 organisms per generation. And if it's up here, it's going to spread. And what this means is that if these are your rules for engineering complex ecosystems, well, you're violating this one, and you are really violating this one. You cannot safely run a field trial of these things. So if we're talking something like malaria, and there's a great team called Target Malaria based at Imperial College that is working to do this to malarial mosquitoes in Africa. The malaria, in the time I've been speaking, it's infected 25,000 people and killed probably 30 kids. That's a problem where every possible ecological side effect combined almost certainly isn't anywhere near as bad as malaria. And so if all the African nations can agree on that and go ahead and do it, that's great. But we don't like tick-borne disease here, but it's not that big of a problem. You probably don't want us using this trick on the white-footed mice, because in principle it's possible, right? In this case, the edited gene you want to spread would be the antibodies. We could use a CRISPR gene drive to spread it to all of the mice on the mainland. It would affect Canada and Mexico too, so there would be some international diplomacy required. But even if it's worked on the Vineyard in Nantucket, I don't think so. I just don't think people are going to go for that, especially because a lot of areas where the mice live don't have that much tick-borne disease because they don't have very high deer populations, for example. We have it worst here. So this is my mea culpa. When I first thought of this CRISPR gene drive, I thought it was pretty great, right? You only see the upside when you first think of something. And so I was thinking, oh, it could do all these sorts of things. No, it won't. It's just way too invasive, especially conservation, just not going to happen. I can think of maybe 4 applications for the full-blown version. Maybe 4. So if gene drive is going to be an option, what you want is something where you can release some mice on Long Island, and it will spread to all the mice on Long Island and stop. That is, you want local gene drive. In fact, you want finer resolution than that if possible. You want it to be only a part of Long Island, a community that says yes. So how do you do that? Well, the problem with the full CRISPR gene drive is it has everything it needs to copy itself. So instead, separate the components of the drive system on different chromosomes. So here's the instructions for cutting, and here's your edited gene and the actual nuclease gene. Here, the instructions cause this to be cut and copied, but they're not copied themselves, so they never increase over time. A increases, but not very powerfully. So in given natural selection, this will be wiped out too quickly. You'd have to release too many mice. That is, it's better than the no-drive version that we're using on Nantucket and the Vineyard, but there's probably at least 1 billion mice on the mainland. You'd have to release far, far too many. But if you instead add a C element, now C has the instructions for cutting at B, B has the instructions for cutting at A, then you get the same sort of effect, but of course you get amplification. So we call this a daisy chain drive because it's just like a daisy chain in terms of how it works, serially dependent elements. Now C never increases, B increases when C is present, and of course A increases whenever B is present, so you get amplification. But, so you can think of it, and again, apologies because I know you're an audience of physicists, but most people think of it as genetic fuel for a rocket, multi-stage booster rocket. And while you have one, then you lose a stage, and then you lose the next stage, and so on. And you get different altitudes, in this case frequency in the local population, note the log scale. And the neat thing about it is the more daisy elements you add, effectively the more stages you add to the rocket, except your fuel actually doesn't cost you anything, which is sort of nice. So it becomes progressively more powerful. And how much powerful is that? If you have 90% homing rate, 90% copying, so this is the very, very low end for the mosquitoes, but high for the fruit flies, you can release 1 daisy drive organism per 50 wild ones with a 5, 6, or 7 element daisy chain. And in 20 generations, it will have spread immunity to, say, your entire local mouse population. For the mice, that's 4 to 5 years. Not that long. If we can get it up to 98% homing, then with a 7-element drive system, you're looking at 1 per 500. So this becomes very cost-effective and potentially very feasible for use all across Eastern North America, any community that wants it. So there's other ways to do it, of course. You can also just scatter copies of B all over the genome. And as long as you have at least 1, then A gets copied. That works as well. And so here's another way of presenting it. And we call this a Daisyfield because you're just scattering daisies all around. And you lose half of your daisies every generation you mate with wild type. So however many you put in, You have that number every generation, and drive happens as long as you have at least one copy. It's just another way of doing the same thing. Daisy chain versus Daisyfield. So these are promising, and we have preliminary data that's very preliminary in worms showing that it works. But it's first worth noting that this sort of requires foreign DNA. And here I have to go in a little bit into how biologists can't agree on what a species is. People think that species are in these arbitrary boxes, but they are arbitrary. That's the point. We don't actually have stable rules for what is and is not a species. Are lions and tigers different species? Well, if you put a male and a female in a cage together in a zoo, you will get either a liger or a tigon, which is an interesting biology element because the growth signals for in utero development are different in tigers and lions, so which one is the mother and which one is the father matters. But the point is they hybridize, and yet we would say that they're different species. So then they sort of hedge that by saying, well, they don't naturally hybridize in the wild, which is weasel words. We don't have a good notion of what a species is, and DNA exchange across what we do call species occurs all the time in the wild. Every sweet potato is a GMO because nature did it. At the dawn of sweet potato domestication 8,000 years ago, there was a bacterially mediated gene transfer event into the sweet potato that moved DNA, and it worked exactly the same way that agricultural scientists most commonly make changes to plants. Just nature did it. Exactly the same way, interspecies gene transfer. And how many of you have eaten beef in the last week? Any beef? Come on, raise your hand. Did you know that on a DNA level, the cow genome is 25% snake? It's true. A couple million years ago, somehow a virus infected an auroch that had previously picked up DNA from a snake. And in that virus was encoded another form of gene drive, a so-called jumping gene. And in the cows, which didn't have any means of suppressing it, it jumped until it was a quarter of the cow genome. So cows are 25% snake. So given that, the notion of foreign DNA is really problematic. But in this case, it depends on, again, what do you call foreign? Half of your cells are bacterial. That is, the bacteria that live in your gut and on your skin, without which you do not function very well, are equal in number to your own cells. And if we were to take a mouse and just extract all the DNA from the mouse, we would have everything we need to make this Daisy drive system. But the CRISPR components come from the bacteria in the gut. So you blend up a mouse, you do get the DNA you need, but it's not from the mouse cells. So does that count as being from a mouse? I don't know. But the reality is we can't do it without using those bacterial DNA. So if people want it, then they will choose to do it. But it's important not to somehow say that we can do it using only pure DNA from mouse cells, what people would conventionally call it. Last problem. I assume there's multiple political jurisdictions on Long Island. What happens if one decides they want to do it and the neighbors say no? How do you get around this problem? We're actually seeing this on the Vineyard as an issue because there's 6 towns on the Vineyard, and they can't even agree if they want artificial turf or grass for their high school. What about engineered mice? Remains to be seen whether they care about tick-borne disease that much. Whereas Nantucket is a single polity, and so working with Nantucket, everything goes very smoothly, and they just have an up or down vote, and that's that. Whereas at the Vineyard, with the version that we're using for them, no drive system, all of them have to agree, because the mice don't know political boundaries. You can't tell a mouse, you can't program a mouse genetically to not go over an arbitrary, invisible human line. So that would seem to be a profound problem for this. The way to solve it, ideally, would be to make the engineered organisms, in fact, the genes comprising the engineering, able to take a quorum vote. Because if they're in the majority, they're probably supposed to be there. That is, we released a lot of them there. They're probably supposed to be there. Whereas if they're in the minority, then they're probably not supposed to be there. So we want to engineer natural selection so that it favors them when they're in the majority and punishes them when they're not. That way, when you have an engineered mouse in the desired community, it's happy and stays there and is likely to stay engineered. Any wild mice that come in will be selected against. But any engineered mice that wander out and mate with a wild mouse, they're going to be in the minority in the neighboring town, and so natural selection will stamp them out. How do you do that? Well, to accomplish a genetic quorum, you want to go to these, what we call haploinsufficient genes. These are genes where you need at least 2 copies to survive. That is, most genes you can get by with just 1 copy, because you just need 1 copy of a gene that makes a functional protein. Haploinsufficient genes are so essential that you need both copies to be functional, or you're dead. So suppose you take 2 such genes, and you swap their positions. Now, when this organism mates with a wild one, the offspring will inherit 2 swapped, 2 original, or 1 of each. The problem is, if they inherit 1 of each, then they're missing a haploinsufficient gene, and so they're dead. What this means is that it's very bad to be a hybrid. Because if you're a hybrid, it doesn't matter who you mate with, half of your offspring are going to die. That's a huge fitness penalty in nature. That's really strong selection against it. So whichever allele is in the majority, in this case, is going to win. That is, if most of your neighbors are swapped, most of your potential mates are swapped, so you have an advantage if you're swapped. Whereas if you're wild, in that case, most of your potential mates are swapped, and so your offspring are probably going to be hybrids, and so you're going to be weeded out. So this is a way of accomplishing this genetic quorum. And what's neat about it is this is something we can spread with the Daisy drive, because whichever form of Daisy drive you use, if you have Daisy elements, while they're there, they ensure that everybody inherits both swapped copies, no matter what. But of course, only half of them will inherit the Daisy element. And when you put this all together, you end up with a situation where the engineered Daisy drive element ones, the circles indicate that they have a Daisy element. Without the circle, but yellow means that they're still engineered, or in this case, immune. If you release the Daisy drive here in this city that wants it, then it will take over here. But when they invade over here, then it will be strongly selected against. And so this population, the adjacent town, will remain wild type. So this is what we hope will be around that solution, to this political quandary, wherein one town really could engineer its organisms, and most of their organisms would stay engineered, but the neighboring town which didn't consent would stay largely wild type. And I should also note that given that we opened this arguable Pandora's box about now we can edit wild populations, this same trick also, theoretically, at least the models say, lets us restore any population to wild type. Because the neat thing about CRISPR is it lets us target any DNA sequence we want. And if you can make a find and replace, that means you can run a find and replace again. So if you don't like the results, you can build another drive system that overwrites it and changes it to something different. Now, if they all spread indefinitely, that doesn't help you that much. But if you use a Daisy to overwrite the local copies, and it spreads these swap genes along with it wherever it goes, it can spread the swap genes, link it to overwriting the engineered genes, then all unwanted engineered genes would be overwritten, and then you'd just have a population that has this engineered swapped haploinsufficience. And then you just release wild types. They're in the minority. Every last copy will be eliminated because the selection gets stronger and stronger the more you're in the minority. So we think that this will allow us to accomplish what we call as a Daisy restoration, which will restore any engineered population, no matter what it is, back to wild type. And that's critical because with CRISPR gene drive, it's a technology that in principle, one scientist can build one if they know how. There's several thousand people in the world who know how to engineer all the flies in the world if they wanted to. So we need a way of allowing everybody else to decide, no, we really want this species to stay the way that we want, and not let random rogue scientists control that. So Daisy drive might be a way to prevent tick-borne disease on Long Island, but it's going to be at least 10 years out. And that's just because we got to do Nantucket and the Vineyard. We got to do the field trial first and on the uninhabited island, and then Nantucket and the Vineyard. In the meantime, if things are looking good, then we will build the Daisy drive in the mice to spread the same antibodies. So we'll hopefully have the field trial on uninhabited island done by the time we have results from Nantucket and the Vineyard. But that would mean for Long Island, you're looking at about a decade out. So I'm sorry to say that I have no answers for you whatsoever for the next 10 years, other than please, please, please spray permethrin on your shoes and your boots and your pants and do tick checks. every time after you go in the woods. Maybe someday we'll be able to fix it with engineering, but not quite yet. And tremendous thanks to everyone in my group for lots of work on both gene drive and tick-borne disease and lots of other things, to our funders for their support. I'd like to highlight the courage of my students in particular, because I'm at the MIT Media Lab. My incentives are very different from most scientists. I'm encouraged to be provocative and suggest that we change sacred systems and generally rile people up. And so for me, going out there and saying, hey, we should pre-register all of our experiments, this may not sound quite as radical to some of you in physics, admittedly, but in biology, this is heresy, because it invites everybody to scoop you. Just run the same experiment, publish first, get all the credit. So I can do it, it's okay for me, but my students are really the ones risking their careers just by joining my lab and agreeing to pre-register all of their experiments if they involve gene drive, just because we believe it's the right thing to do, and someone has to lead the way. So special call out to the heroism of my students, and thank you once again for the invitation, and all of you for your attention. Yes, so the question is, all of this, what I showed you assumes a static genome, that there's no variation in the target population, that the things that you are carrying with the drive are not going to mutate, and in particular the guide RNAs that might allow it to target another site. So CRISPR itself, it's a great question. So CRISPR itself is extraordinarily specific. That is, if we use the high specificity variants, it really will not tolerate cutting any site other than one that is a perfect match. But there is nothing stopping the guide RNAs from mutating. Now, you should have selection against that if they are cutting something else, because it can't actually copy itself somewhere else in the genome unless it also has the homology. So if it just cuts it and there's no homology, then it will just break whatever that target sequence is. And that's probably costly, so we would actually expect natural selection to act for drive systems that continue cutting their designed target site ever more efficiently. The other problem is the resistant allele stuff that I showed. That assumes that you're just using one guide RNA. If you want it to be evolutionarily stable, that is to spread to every member of that population, you have to ensure that all the possible resistant alleles are more costly than your drive system. So that means you have to target many different sequences, so you're absolutely certain that at least one of them is going to cut. And evolution has no opportunity to mutate all of them, because it would have to mutate all of them at once. And all of those have to target a sequence that is important enough for fitness such that if it just deletes all of them, then that's more costly than the drive system. So if you do all of that, then gene drive is evolutionarily stable in the sense it's not vulnerable to these drive-resistant alleles. And it shouldn't evolve the guide RNAs to cut anywhere else, because that should be costly to the drive system. But admittedly, that's a fairly weak selection and relative to the time that the drive spreads. So we're not positive that. We still need to do more modeling on it. And let's see, the last thing is the antibodies that we're spreading in this example. There is no selection pressure for keeping those functional. The mice don't get disease. They don't need them. Now, keeping the mice from being infested with ticks might be a fitness advantage, very slightly, but I rather doubt it, because the existing studies suggest that there is no effect whatsoever, even if you put dozens of ticks on a mouse. It doesn't seem to impact its reproductive success. So we don't think that that will change, which means that there's no pressure for those antibody genes to remain functional, meaning that they will likely break over time. So how long is that? At least decades, because the fitness cost to the mouse is pretty small. You're just asking it to make antibodies. It's already making hundreds of other antibodies, because that's the way its immune system normally works. The levels required for immunity is quite tiny. So we don't think it's going to be particularly costly, but it will break over time. So it's probably going to last decades to centuries. If it's decades, then you might have to use a different Daisy drive to overwrite it, or just add fresh copies every 50 years. That would probably be okay. I don't really worry about it if it's over centuries. But it's a great question, because we have no experience engineering systems that are expected to evolve outside of our control. We've just never done it before. You know, yes, we engineer crops for farms, but we control the environment there, notably to spray it with herbicide. This is something that will evolve in a wild population outside of our control, barring us releasing another one. So it's something that we really need to be careful before we think about doing it, especially if we're going to use the full-on CRISPR gene drive. And lastly, there's the question of species again, in that whole fertile hybrid thing. So we're interested in building a daisy suppression drive to reduce the population of the southern house mosquito, which is invasive in Hawaii and killing all of their endangered birds by spreading bird malaria. But the southern house mosquito can hybridize with the northern house mosquito. In fact, there's a lot of gene flow between them, and it's the northern ones that we have around here. So we're only doing daisy drive, but if someone did a full CRISPR gene drive, you would expect it to spread between them if the target DNA sequences are there in both. They might not be. But we need to know, but what if, even if they're different, if they're only slightly different, will the guide RNAs evolve to spread into the species that they couldn't previously do? We would expect there to be pressure for them to do so because then they would have a fitness advantage. And so we're studying these systems in worms because we can grow 1 billion worms in the lab. And evolution is a numbers game. So you have to grow as many organisms as you think you're going to alter, or more, in order to have a chance of picking up an event like a guide RNA mutating so that it can spread into some related species. So there's a long way to go before we do any of these gene drive stuff in terms of the safety on that angle. Yes? So we're very adamant that our initial experiments not involve gene drive because A, we still need to run all of those worm experiments to work things out. Daisy drive is still very new. And we want to give the islands some chance of something first. Also, most of the citizens we've talked to would prefer that we not use any foreign DNA, even if it's from the bacteria in their guts. Interestingly, almost anyone with any kind of technical training says, wait a minute, you could release fewer mice, it'd be more efficient, it'd be less costly, it'd be faster. Why not? And people without technical training largely say, no, let's just keep the mice, 100%. It is what it is. So and the other problem is that my personal name is very much associated with gene drive. If you Google my name, you will see lots of stuff about gene drive. So everyone by default assumes that our tick-borne disease project on Nantucket and the Vineyard will use gene drive just because they think of me as the gene drive person. So I have to emphasize, so every journalist who writes a story about it always initially puts in that we will use a gene drive on Nantucket and the Vineyard, and we're not. So I have to emphasize that every chance I get. Yeah, so for these experiments, we haven't done any form of gene drive or even daisy drive in the white-footed mice. So our white-footed mouse project is still, it's a year old in terms of for how long we've been doing any kind of experiments at all. What I'm laying out here is if you don't use something like a daisy drive, or hopefully something even better that someone will invent in the next few years, what we're doing on Nantucket and the Vineyard just would not work here on Long Island. There's just too many mice here. It's too big. So we would need something like a daisy drive. And so I was laying it out because if it's going to matter to all of you and your neighbors and families here on Long Island, then we will need a daisy drive to spread the same antibodies that we're using on Nantucket and the Vineyard. But over there, we are not using any form of gene drive. And I will pretty strongly oppose anyone who suggests using the standard self-sustaining CRISPR gene drive, just because I think that would be very reckless. Now, if 100+ cities all over the country, up into Canada as well, decide to use the daisy drive version, and it works and it's working great, and then people decide, hey, I want to hike the Appalachian Trail with no risk of Lyme disease, then maybe we can talk. But again, you want to start local and scale up before you do something like edit an entire species.
Behavioral or social issues about introducing your laboratory mice and competing with the wild ones?
Great question. Behavioral or social issues or general fitness of lab-raised mice when released into the wild. So here we're sort of adopting the cicada strategy. That is, just come out in such overwhelming numbers that all of the predators eat their fill of your mice that don't know how to hide from hawks and cats and stoats and so on. And there will still be so many that they will be fine. The other trick is that we are releasing them, planning to release them at the end of winter. So all of our mice will be fat and well-fed and happy, and so they'll be hungry for a while because there's not much food out there, but they'll be better off than the wild mice, which will be thin and starving. So the extent to which those will counterbalance the fact that our mice will not know how to look out for hawks and so on, we don't know. We might have to release more than we anticipated in order to overcome that, but it would probably be a matter of release the number that we expected and then sample how abundant the gene is, and then maybe have to do it again the next year. Yeah, we just don't know.
You mentioned when you're talking about CRISPR that you might use with higher specificity. Are you looking into building any safeguards other than that towards making sure it targets the right thing? Guide RNA built in, and I know some.
Yes, so there's different ways of doing higher specificity CRISPR. There's some where the newer ones, the nuclease itself has been engineered to require to be intolerant of additional mismatches. You can also break one of the DNA cutting domains so it only cuts one strand, and then you require two cuts, one on either strand, in order to generate the double strand break to catalyze it. We also might be able to do it using only the nickase version that only cuts one strand, because it's much less efficient. But if you have a bunch of guides all cutting the same strand, that's biased towards copying rather than jamming the ends together, so it might make it for a better gene drive. There's lots of different ways of doing it. Most of the images that I showed use just one guide element for simplicity of viewing. In reality, we would almost certainly use 5 at a minimum for any given element. And it's a little bit more complicated. The Quorum systems require a little bit even more than that for reasons I don't have the slides loaded in this deck to explain. But in general, we would use whatever is the most efficient, precise version available at the time, and we're going to need to experiment as to the nickase's approach versus the full nuclease and what would be better. Yeah, great question.